All right, Team Science, we're going to keep talking today about how cells control when and how much RNA is made. When we're talking about eukaryotes, we might be talking about how the cells decide which genes to turn on. Am I going to be an eyeball or am I going to be a toenail? Your cells determine that based on which genes are turned on. Now, in a prokaryote, though, there's less specialization. However, the prokaryotes, like eukaryotes, need to be able to respond to their environment. And we're going to talk today about how prokaryotes can use operons to respond to their environment. And we're specifically going to look at the lac operon, one of the most classic gene expression regulation systems that's ever been studied. When we think about the lac operon, we need to think about two different scenarios. The presence of lactose and the absence of lactose. And we're going to start by thinking about what's happening to an E. coli cell in the absence of lactose. Now over here on the right, you can see we've got our operon. That is, we've got a promoter that contains an operator region. Remember, this operator is like a consensus sequence. And then we have multiple genes that are part of the operon. And in this case, the names of the genes are LAC-Z, LAC-Y, and LAC-A. But in the absence of lactose, we don't actually have transcription of these genes at all. And that's because of a separate protein. This is our regulatory protein. You can see over here that the regulatory protein has its own promoter region and its own gene coding region. The name of the regulatory protein in the lac operon is LAC-I. I probably for an inducible system, the lac operon is a negative inducible operon. But this is a repressor protein. Okay? And in the absence of lactose, you can see that this protein has a nice little shape that allows it to wrap around the DNA. And it's going to bind, if we zoom in down here on the bottom, our repressor protein, our lac repressor, LAC-I protein, is going to be binding to a region just adjacent to the promoter. And this is the consensus sequence, and it's often called the operator region. Okay, so this is the consensus sequence that the LAC-I repressor is looking to bind onto. Now you can imagine that if the RNA polymerase were trying to travel down this gene to transcribe, it would physically be blocked by this LAC repressor. So with no lactose, the LAC repressor is bound, RNA polymerase is blocked, so we have no transcription of the LAC-Z, LAC-Y, and LAC-A genes. Now let's consider what happens in an E. coli cell in the presence of lactose. All right, we've got our E. coli. Now we've got lactose present in the cell. We've still got the LAC-I gene over here on the left. We're still transcribing and translating a protein. And this repressor protein is still trying to do its job in the cell. It's still trying to bind to this operator region of the LAC promoter. However, when there's lactose or allolactose, in this case, present inside the cell, the allolactose is able to bind to the repressor protein, and you can see this changes the conformation of the protein. It no longer has that shape that will allow it to bind DNA. So the repressor is not on the DNA. Instead, RNA polymerase is able to move through here, and first we'll transcribe an mRNA, and because we're in prokaryotes, instantly that's going to also be translated into 3 different enzymes. So you maybe recall we've got the LAC-Z gene that encodes beta-galactosidase, which is an enzyme. We've got the LAC-Y gene, and that encodes permease. And then last but not least, we have LAC-A, and that encodes a protein called transacetylase. Permease is important for helping get lactose across the cell membrane. So lactose comes inside the cell with the help of the permease protein. And once inside the cell, lactose can be turned back and forth between allolactose and regular lactose by the enzyme beta-galactosidase. Beta-galactosidase can also help break lactose down from its disaccharide form into these two monosaccharides, and this is what the E. coli are going to use up and metabolize for ATP. So just to clarify, we've got this negative inducible system that E. coli only turn on, they only induce expression of this operon when there is lactose in the cell. So when we have lactose around, whether it's allolactose or regular lactose, when the cell senses the presence of lactose, it's able, and it's sensing it through this transcription factor here, it's able to turn on gene expression to allow the cell to metabolize the lactose and use it for food energy. We don't need this system all the time. That's why it's inducible, normally off and induced to turn on in the presence of lactose. I want to talk a little bit more about the components of this system, and to do that, we're going to need to talk about some historical scientific research. So this research was started by Jakob and Monod, and we're going to call it a partial diploid experiment. Remember that E. coli and most bacteria are haploid. That is, they have one copy of their chromosomes, or their single circular chromosome. And in fact, if we draw an E. coli down here on the bottom, I'm going to draw its DNA over here. It's a big circular chromosome. That's our chromosomal DNA. So we know that somewhere on this chromosome is a copy of the lac operon. Jakob and Monod added in a second copy of the lac operon. So I'm going to draw that second copy of the lac operon on a plasmid. And this plasmid is actually called the F' plasmid. So that's what this is referring to. And on that plasmid, we have a copy of the lac operon and not much else. That's why this is just a partial diploid. It's not diploid at all regions of the chromosome. It's only diploid for the lac operon. So now this E. coli, you could think of this as a genetically modified E. coli carrying extra DNA. This E. coli has two copies of the lac operon. It's got one copy of the lac operon on its chromosome and one introduced region of DNA that also contains the lac operon. And just like we were looking at a moment ago, we've got the repressor protein over here. We've got the promoter and operator regions. And then we have the two genes we're really going to focus on, LAC-Z and LAC-Y, that encode beta-gal and permease. And we have all of those same components also represented on this F' plasmid, the second copy of the lac operon. The first thing that Jakob and Monod determined using this partial diploid system, so partial diploids tell us that the repressor proteins, that is, our transcription factors, are transacting. So trans, remember, means across, and you can also think of this as being global. Repressors or transcription factors act globally. It's kind of nice, transcription factors transacting. The way that Jakob and Monod use the partial diploid system to tell us this is that up here we've got our chromosomal version, and down here we've got our plasmid version of the lac operons. And you can see up here in our chromosomal version, we've got I minus. This I minus, this lac I minus, does not encode a functioning repressor. So we're going to make, like, we have the absence of a repressor here. However, we can still make a repressor from the functioning I plus repressor gene that's found on the F prime plasmid. Now, what Jacob and Monod wanted to ask was, without lactose, do we have expression of beta-gal and permease? Because if you don't have a functioning repressor over here, you might expect that these genes could be transcribed. What they found was that no, even though this version of the lac operon doesn't make a repressor, this version of the lac operon does, and that repressor is able to act on its own operator region, but it's also able to travel and act globally and repress transcription nearby and also on the chromosomal version of the lac operon. Repressors or transcription factors can travel. They're transacting. The next thing that Jacob and Monod used our partial diploids to determine is that operator regions are cis-acting. And remember that cis means same, but it can also mean local in this case. So the other thing to remember is that an operator, this is a consensus sequence. This is a sequence of DNA, okay? The DNA isn't traveling anywhere. It can only act locally. So in this case, we've got a functioning repressor on our chromosomal version of the lac operon. And then we've got our plasmid version of the lac operon still making, both of these guys making a functional repressor. But what's different is that on the chromosome, we have an O plus, an operator with the normal sequence. And on the plasmid version, we have an OC. And this is called a constitutive or constant operator. And this is a mutation. And the repressor cannot bind this altered operator. Let's talk a little bit more about what that actually means. Let's imagine that the sequence of this normal operator is TA, TA. Okay? And that's the sequence that the repressor protein goes looking for to bind. However, in our constitutive operator, the TATA has been mutated into TCCA. Well, that's not the same sequence at all. When the repressor protein comes looking for this, it's going to keep right on looking because this doesn't look like the correct operator sequence. So, Jacob and Monod were able to determine that without lactose, we still have expression of beta-gal and permease. And that doesn't come from the chromosomal version because here transcription will still be shut down by the repressor binding. However, over here with our constitutive operator, we can have transcription all the time because the repressor is never binding to this region of DNA. It just keeps on looking for the consensus sequence. So the operators they were able to determine are cis-acting. They act locally to affect expression of the genes which are just nearby them. So we only affect expression of these neighboring genes when we mutate the operator. The more distant genes are not affected when we mutate an operator. Last but not least, Jakob and Minot used the partial diploids to show us that the repressor protein, or the transcription factor, contains a lactose binding site and also a DNA binding site. Okay, so we know that this repressor needs to be able to bind to the operator region. But importantly, there's another component of the repressor protein, and that is its ability to bind lactose. So the Jakob and Minot used a different mutation. Rather than an I- mutation, here we have a super repressor. So IS is super repressor, a suprapressor. Okay, and this super repressor is still able to bind to DNA, but it can never bind to, in this case, the inducer, which is lactose. Okay, we're calling it an inducer because it induces gene expression. Now, in the chromosomal version, we've got the super repressor. In the plasmid version of the lac operon, we've still got the normal lac I+. And that repressor protein is able to bind to lactose, the inducer molecule. That means this repressor is no longer able to bind to DNA. So we're able to have transcription of beta-gal and permease. So in the presence of lactose, we expect transcription of beta-gal and permease. And of course, in the absence of lactose, we expect no transcription. What will occur if we add in a super repressor? That is, a repressor that is blind to or can never bind to the lactose inducer. Well, what Jakob and Minot ended up seeing was that without lactose, the super repressor shows no transcription of beta-gal and permease. And in the presence of lactose, we have no transcription. And that's because this repressor protein, the super repressor, is able to bind here and block transcription. And because we know already that repressors are transacting, it can also travel over to this copy and prevent transcription of the plasmid lac operon as well. So we've got the transacting ability of our repressor. It's got a DNA binding site that's still working, but now the lactose binding region of this protein is broken. So that's what the super repressor mutation is. The last thing I'd like to do is for us to think about some problems considering these DNA sequences, these genes in the lac operon. We're going to be using these codes to represent the DNA we're interested in, or the genotypes that we're interested in, using the mutations that we just talked about. So under normal circumstances, we have a functional repressor, a functional operator, LAC-Z, and LAC-Y. Okay? So in the absence of lactose, we expect for this repressor to bind and inhibit the operator. So we're not going to have any beta-gal made from LAC-Z. So beta-gal comes from LAC-Z. Permease comes from LAC-Y. But in the presence of lactose, the repressor will fall off the operator. We'll be able to see expression of beta-gal and permease. In this second scenario, we actually have a Z- mutation. We're not affecting transcription at all. So in the absence of lactose, we still should have no expression of beta-gal and permease. And in the presence of lactose, permease will still be made. But because we have a Z- mutation, we won't ever be able to make beta-gal. Remember, beta-gal is encoded by the LAC-Z gene. So if we have a mutation in LAC-Z, we're not going to make a functional version of beta-gal. In our third scenario here, we've got the super repressor. So it's a repressor. That means in the absence of lactose, we know we're not going to have any transcription or translation. And in the presence of lactose, we're actually still not going to have any transcription or translation because the super repressor is blind to lactose. The super repressor doesn't see lactose. So even though we've added it into the system, doesn't matter. The super repressor keeps repressing. Let's consider now our fourth scenario. We've got a constitutive operator, that is a mutation in the operator. And we still have our nice repressor protein. Well, the repressor protein is going to try and bind to this operator. However, because the operator sequence is mutated, it's not going to work. Even though the repressor protein is going to try and bind, we're not ever going to have repression. We're always going to have expression of beta-gal and permease without or with lactose, because this constitutive operator essentially renders our repressor inactive. Let's consider then this related case where we have an I- mutation. So the IS mutation we just looked at is a mutation in the lactose binding region. The I- mutation is a mutation in the DNA binding region of the repressor protein. If the repressor is never able to bind DNA, it's never able to inhibit the transcription. So we're always going to have transcription, even in the absence of lactose, if our repressor protein is broken. Last but not least is a fun one to consider. We've got both the super repressor and the constitutive operator here. Which is going to win? We've got a super repressor who's always trying to turn off gene expression. And we've got a constitutive operator that is a mutation in the operator so that there's a different DNA sequence there. Even though this super repressor is going to be trying hard to inhibit transcription with or without lactose, if the constitutive operator has changed its sequence or has a mutated sequence, then the super repressor won't ever be able to bind. The constitutive operator wins out over the super repressor. This is actually an example of epistasis. And I'm not going to talk any more about that, but if you're interested in epistasis, it's a really cool phenomenon. So the mutation in OC, the mutation in the operator, sort of just like we drew up here, is going to be blocking the effect of the repressor, no matter what type of repressor that is. Now I'd like us to think about a couple of problems involving partial diploids. Just to remind you, here's our normal lac operon situation at the top here. In the absence of lactose, we have no beta-gal, no permease. But in the presence of lactose, we're able to make both beta-gal and permease because this repressor protein will fall off the operator and allow transcription to occur. If we consider our partial diploids now, first we're looking at the chromosomal region, and then F' indicates we're looking at the plasmid region, that second copy of the lac operon. And you can see in our chromosomal version of the lac operon, we've got I+, O+, Z+, Y-. So that's a mutation in permease. And then in the plasmid version of the lac operon, we've got I minus, O plus, Z minus, Y plus. Okay, let's first consider in the absence of lactose. When I look at this, the first thing I see is this I minus. If we have an I minus, doesn't that mean that we're going to have expression of this all the time because there's no repressor here? Well, remember, this repressor protein is actually able to transact, that is, act globally, inhibit the operator right next to it, but also inhibit this more distant operator. So I'm going to say in the absence of lactose, we're going to inhibit expression of both lactose and permease here and over here. And then in the presence of lactose, we're going to allow transcription. We're going to turn off this in repressor protein. We're going to have transcription of both beta-gal and permease. Okay, let's think about this second example here. We've got the chromosomal version, which has I plus, the constitutive operator Z plus Y minus. And then on the plasmid version of the lac operon, we've got I minus, O plus, Z minus, Y plus. So this is pretty similar to what we saw up above. Just because we have an I minus, this I minus doesn't do anything. Instead, the I plus, the repressor protein can travel over and act in a trans fashion to inhibit expression of this version of the lac operon. And of course, it can inhibit, this repressor protein can't inhibit locally because why? Because we've got this constitutive operator. The constitutive operator, remember, is something that the repressor proteins can't find, can't bind. That means in the absence of lactose, this part of the lac operon is going to be totally inhibited just like normal. But this part of the lac operon, this copy of the lac operon, will be transcribed. In the absence of lactose, we're going to be able to make beta-gal, Z+. We're still not going to be able to make permease, though, because this is a Y minus. Remember, permease is lac Y, lac Z is beta-gal. Then, in the presence of lactose, now this inhibitor will go away. We'll be able to make beta-gal from this gene, and we'll be able to make permease from this gene. One more example for us to think about. We've got on the chromosomal version, we've got a super repressor, O plus, Z plus, Y minus. And on the plasmid version of the lac operon, we've got a constitutive operator, Z minus, and Y plus. So what's going to be happening during the no lactose situation? Well, this super repressor is going to be repressing the operator found on the chromosomal version of the lac operon. And actually, it's going to be repressing this without lactose and also with lactose because it's a super repressor. What about over here? How is this going to be affecting the plasmid version of the lac operon? Oh, well, you can see that we've got a constitutive operator here. And we've already discussed that if there's a mutation in the operator, this super repressor isn't going to be able to block it. That means even in the absence of lactose, we're still going to have expression of this version, this part of the lac operon. But again, we only have a functional copy of permease. So here, we'll be making permease even when there's no lactose around. But we won't be making beta-gal. And then in the presence of lactose, this is still going to be inhibited in the presence of lactose. So we're actually still just going to be transcribing from this F prime plasmid version of the lac operon. This super repressor is going to be constantly repressing the functional operator that's found on the chromosome. So we don't actually see a change here in the presence or absence of lactose. So I hope that helps you think about how these partial diploids can work together to give us more information about how the lac operon is regulated using, we've got different transcription factors, we've got consensus sequences, and we've got lots of mutations that all come together to help us understand how this gene expression is regulated.